Welcome back. It's my distinct pleasure to introduce our afternoon speaker, Dr. Lina Shehadeh from the University of Miami School of Medicine. Dr. Shehadeh received her PhD in bioinformatics from Florida Atlantic University, and she has been at now for how many years? 15 years. She's very well recognized in the field, particularly in the role of osteopontin in cardiac disease. And she also works in a lot of cutting-edge therapies such as iPSC differentiation, gene therapies, and so on. And she has, I guess, she's well known for her work in osteopontin in particular. I believe she has a, NIH R01 grant on that topic. And she's also well recognized by several other facilities, especially locally here with the Miami Heart Research Institute, where she's been very well recognized as well. So again, it's my distinct pleasure to introduce our afternoon speaker. Please join me in welcoming Dr. Lina Shehadeh.
Thank you so much, Sharon, for this very kind invitation. I'm very honored to be here. I think this is an amazing platform to spark collaborations between FLU and I have a big shoes to fill after speaking after Dr. Yuski, but I'll try my best. Please be engaged. Feel free to interrupt me and ask questions anytime. You don't have to wait till the end. I was going to talk about a different topic, but after listening to Dr. Yutsky, meeting most of you here by the posters, I thought I'll talk about my other big project in the lab that's related to calcification and atherosclerosis. But I'd like to give you an idea of what I was going to talk about, and you feel free to ask me after about it or email me. And that's the topic of heart failure with preserved ejection fraction. So heart failure is the #1 killer disease in the world, and the very common form of heart failure is heart failure with reduced ejection fraction, or HFrEF. Over the past few years, another form of heart failure emerged, and it's called heart failure with preserved ejection fraction, HFpEF. And it's in 50% of heart failure cases, and it has been unrecognized because doctors rely on ejection fraction as the measure of cardiac function. So ejection fraction is how much blood is pumped out of your blood, how much blood is pumped out of your heart. And if it's reduced, then it's a sign of heart failure. If it's normal, then the doctors think of other sources of disease. And it turns out that HFpEF is, like I said, in half of heart failure cases, and all of HFrEF medications do not work on HFpEF. So it's a very hot topic. NIH is very interested in developing models for HFpEF. You guys here, in terms of engineering cellular platforms for HFpEF, it's very timely, very novel, and this is actually one of the reasons why I jumped into HFpEF, because I saw there's a niche for it, there's a need, NIH wants to fund it, and I think it has a lot to do with stiffness of the heart. Even though the heart pumps blood normally, the heart is stiff, and there is abnormal filling pressures of the heart. This disease is complex because it's usually comorbid with diabetes or obesity or renal dysfunction. So not every HFpEF patient is like the other. It's complicated. Like I said, it's an opportunity for you all to think about how to benefit this field, the ones of you who are interested in cardiovascular applications for your engineering ideas. That being said, I'll talk about the other big project in the lab that's been actually going slow, but going for sure. And the reason I prioritize HFpEF in terms of my grant applications for atherosclerosis even though my key award was on atherosclerosis, is that this is a disease that's been there forever. It is the source of cardiovascular problems. High cholesterol, clogged blood vessels is the source of heart attacks and strokes. It's been studied for decades and sort of saturated. The field is saturated, and the progress is very slow. So for someone to say, I'm going to make a huge difference in this field, it's very challenging. Nevertheless, we don't give up on this important topic, and we've been working on it for so many years, and hopefully we'll be able to contribute soon. So I'll be talking about MIR30E and how we got to Osteopontin in this project. So the summary of the key events that happen in atherosclerosis: there is elevated plasma level of LDL cholesterol, LDL for lousy cholesterol, and one of the main most common animal models used is the ApoE knockout mouse, which I've seen several interesting posters using this model. This model has high LDL cholesterol in the circulation, in addition to plaque, and that's accelerated when you put the mouse on high-fat diet. With atherosclerosis, macrophages uptake the oxidized LDL, and they activated, they secrete cytokines and other growth factors. Smooth muscle cells become proliferative and migrate to the intima, forming plaque. And vascular calcification and plaque rupture happens, causing stroke or coronary artery disease. So with this, our focus was on smooth muscle cells because they're main players in this disease. They're really inseparable. All of these events are inseparable. And as you will see, we will get back to high cholesterol level. Again, the data guided us to get there, and we have to follow our data. So this is a classic example of a normal blood vessel versus an atherosclerotic one. You can see in the normal blood vessel, you have a nice layer of smooth muscle cells surrounding the endothelium. These smooth muscle cells are differentiated. They express smooth muscle markers, they contract, they preserve this nice diameter of the blood vessel. However, in an atherosclerotic vessel, you have cholesterol causing insult on the endothelial layer. You have this plaque forming in the subendothelial space where, like we said, smooth muscle cells start dividing, proliferating, and macrophages uptake cholesterol, causing foam cells. So like we said, in a healthy vessel, you have a nice layer of smooth muscle cells. They express the smooth muscle markers. However, with a disease, these become de-differentiated, or they're called synthetic. This phenotype is like, the cells become like mesenchymal cells. They proliferate, they migrate, they secrete cytokines, and they can differentiate into osteoblasts. So hence the calcification that we've been hearing about is very interesting, not only at the valvular level, it's at the programmatic level, at the genomic level, where you have this osteoblast program causing various pathologies at different levels in different tissues. So this is an example of a histology from an ApoE mouse. You can see how in the subendothelial space, you have all of this plaque form, and you see the calcification pointed by the arrows, and you have expression of osteogenic markers like insulin growth factor 2, RUNX2, osteopontin, BMPs, etc. This is a fluorescent image of the same vessels. You can see here in green the osteopontin expression within the plaque. And these are histology from human aorta that we get from explanted hearts. you can see here again, alizarin red, how the vascular calcification is eminent in these aortas. So through various genomic screens, we did microarrays, and my background originally is bioinformatics. I mine data, our own data, and map it with NCBI deposited data. I looked for genes that were dysregulated in atherosclerosis, and I found that this gene, NFYC, was dysregulated in several data sets, so I decided to focus on it. And I found that this gene hosts this microRNA. In fact, it hosts two microRNAs, miR30E and miR30C, in its intron. So we did some preliminary qPCR to look at the transcripts, and we compared the young and the old, and wild type versus ApoE aortas. And we found consistently that the NFYC, the host gene, and the microRNA were consistently down regulated in atherosclerosis and with aging. And we found that the osteogenic markers, again through bioinformatics, it was clear that many of the targets of the microRNA were osteogenic markers. So we looked at osteogenic messenger RNAs that are targets, and we found that they were reversely regulated, meaning the miR was down and these genes were upregulated. So we did functional experiments. We added the microRNA to the smooth muscle cells or to bone marrow-derived mesenchymal stem cells, because remember I told you the smooth muscle cells, when they differentiate, they act like mesenchymal cells. And we found that when we added the microRNA here in red, the proliferation was significantly reduced in the mesenchymal stem cells. And the adipogenic, the fat markers were upregulated. So usually in cells, in stem cells, they have to choose between adipogenic, the fat lineage, or the osteogenic, the bone lineage. So it makes sense that if you push the adipogenic lineage, you suppress the osteogenic lineage. And indeed here, you see that when we add the microRNA, all of these osteogenic genes were downregulated. So this was strong evidence in smooth muscle cells and in mesenchymal stem cells, was clearly convincing that this microRNA suppresses osteogenic differentiation. So what happens with the smooth muscle markers? Remember I told you when the smooth muscle cells become synthetic, they lose their the smooth muscle markers. When you put in the microRNA, as you see here, the smooth muscle markers are elevated. So that's something you do want to program the cells to. So smooth muscle, SM22 alpha, calponin, vinculin, all are clearly upregulated when you introduce the microRNA. So we did microarrays again, whole genome expression levels with the miR and with the anti-miR to screen them down as to what is the primary target. And by converging the data sets, we found that IGF-2, which is also an osteogenic gene, was upregulated with the anti-miR and suppressed with the miR. So we did binding assays and confirmed that indeed miR30E binds to the 3' end of this transcript and suppresses its expression. And the opposite happens when you introduce the anti-miR. The IGF-2 transcripts go up in various cell lines. So this was mesenchymal cells, smooth muscle cells, liver cells, and primary hepatocytes. So this was quite convincing. We also did studies in vitro where in vivo, where we injected the ApoE mice with the anti-MIR, and we collected tissues, and we saw that the IGF-2 transcripts were elevated. We did continue to do functional studies to validate that IGF-2 was the mechanism of function. So if you add miR-30E, you suppress osteogenic differentiation. If you add the IGF-2, which we believe the miR-30E blocks, it blocks the function of the miR-30E. So in conclusion, this study showed that miR-30E and its host gene, NFYC, are downregulated in the ApoE aortas and inversely correlated with osteogenic genes. miR-30E downregulates a panel of osteogenic genes in mesenchymal stem cells also. miR-30E promotes the healthy phenotype of smooth muscle cells and attenuates osteogenesis. IGF-2 is a primary target of miR-30E, and IGF-2 recombinant protein represses the miR-30E effect. So we wanted to, I was very excited to go to animal studies with the MIR, and I talked to some venture capitalists, so that, I found the target, we patented, let's treat, atherosclerosis with miR-30E. And reality check, we get very excited about our work. And translationally, the microRNAs are not close to clinic yet. And why? It's because they go everywhere and you have all sorts of side effects. Things in mice are easier to work. It's easy to cure the mouse. But in humans, you don't want the MIR to go to all of these organs where they're perfectly fine. You're not going to make the MIR go specifically to the aorta, for example, and work on the smooth muscle cells. So what I was advised is why don't you look at the targets of the MIR? If the miR-30E is that powerful, look at the targets of the miR-30E. And IGF-2 was the primary target, but really the protein that we believe was making all of this effect on the phenotype is its indirect target, which is osteopontin. So we started a series of experiments in vivo with the osteopontin, and we crossed the ApoE, the atherogenic ApoE mice, with an osteopontin knockout mouse. Again, going back to the challenge of taking this to a cutting edge level and of novelty, it's challenging. In fact, in 2003, there was a paper published in ATVB showing that osteopontin knockout in ApoE mice rescued the females and not the males. So, and it ended there without any mechanism, without any explanation why this gender effect, which in fact NIH is help us to repeat those experiments and see what we get. We thought we'd let the mice age. We didn't put them on high-fat diet. In fact, we saw the exact same effect. This is the aortas from the females, female ApoE versus female ApoE on osteopontin knockout, and male ApoE on osteopontin knockout. We did all of these quantifications. They're more compelling in the carotids because you get 2 carotids per mouse in the versus 1 aorta, and indeed there was a significant reduction in the total amount of plaque in the carotids. For anyone who's worked with plaque lesion size in ApoE mice, it is very hard to have a significant reduction in plaque. Because, like I said, it's actually easy to add the plaque, but to reduce it, it's very challenging, and to get a significant number with so many ends is very challenging. So this was exciting. This is real. This is why is it in females and not in males, and what is osteopontin doing that is blocking this? Is it at the cholesterol level? We don't see significant differences in the circulating plasma. That doesn't mean it does not affect the uptake of cholesterol. Does it act at the plaque level in the smooth muscle cells? Is it the macrophages? A lot of unknown questions, and that must be answered. Same for the histology of the plaque. You can see that in the females, there is significant reduction in the lesion root area, but not in the males on the osteopontin knockout. So last year we published in JCI Insight in a kidney model that when we knock out osteopontin, we were rescuing a kidney disease, a kidney phenotype. And one of the main findings of that paper was that the Ldl receptor, again, this is low density lipoprotein receptor, the receptor that's responsible for cholesterol uptake, was upregulated in the kidney tubules, as you see here in the sick mouse, and shown in brown. And these kidneys, because they have a lot of this receptor, they have more of the cholesterol, and there's lipid toxicity in the renal tubules. However, with the osteopontin knockout, as you see here, there was in the quantification here, there was significant reduction in the Ldl receptor. So that was exciting. Like, no one has reported osteopontin regulating the receptor for cholesterol. How does it work? How is this? Is it direct or indirect? We decided to venture into that and see if that's related actually to what's happening in the ApoE mouse. This is just to show you when you inject cholesterol in this wild type versus kidney disease model, labeled cholesterol, you can see that because as expected, there's a lot of receptor, there's a lot of uptake of the cholesterol. And when you knock out the receptor, you can extend the lifespan of the mouse. So this is at the essence of this disease. In human tissues from explanted hearts from atherogenic patients, ApoE, osteopontin, and Ldl receptor are highly expressed. Sorry. Oh. About that. Okay, so osteopontin and Ldl receptor are also expressed in tissues in different cell types in the plaque, as you can see here in the endothelial layer and also in the plaque itself, probably in the smooth muscle cells. So Ldl receptor is not just important for the liver, where it's mainly studied; it's also important in the plaque. So where are we with this study now? We developed a series of monoclonal antibodies against osteopontin. Because, like I said, translationally, monoclonal antibodies are more acceptable. There are a lot of FDA-approved drugs that are monoclonal antibodies. We developed anti-human osteopontin antibody. It also recognized the porcine osteopontin. It does not recognize the mouse, that's why we couldn't use it in mouse. Out of the 20 antibodies we generated, one of them had the best KDs compared to the commercial antibody out there. When we treat the liver cells, which have a lot of Ldl receptor with the osteopontin antibody, we were pleasantly surprised that there was an increase, as you see here by the shift to the right in the black line, with the Ldl receptors on the hepatic cells. And that's important for cell cholesterol clearance in the liver. So we did this cholesterol uptake assay in human liver cells, where you put this labeled, fluorescently labeled cholesterol on the cells and see how it affects cholesterol uptake over time. This is live imaging over time, very nice high throughput assay that normalizes the fluorescence intensity over the area of the cells. And you can see that when you compare the DMSO or the IgG treated cells with statin, for example, upregulates the number one drug out there for hypercholesterolemia. And you see here in blue, upregulates LDL cholesterol uptake. The antibody, our antibody here in gray, even upregulates more. And if we do a combinatorial therapy, you can see we got the best results. And so this is exciting and promising, and we're trying to get pharma interested in this because Repatha therapy, for example, by Amgen, anti-PCSK9 therapy is given in addition to statin to improve cholesterol uptake in the livers. And finally, the last piece of data I have for you here. No, I'm doing this time. We did biochemical assays and contacted some biochemists about this to see what does this mean. We did direct binding assays of osteopontin to Ldl receptor, and indeed they do bind with high affinity. And according to the biochemists, you can call this a new receptor ligand interaction. So osteopontin is known to bind to integrins and CD44, not to Ldl receptor. But here we're reporting that Ldl receptor is a new receptor, or osteopontin is a new ligand for Ldl receptor. Where do we go with this? Now we're trying to get funding to do injections in a pig model of atherosclerosis. There's this pig called the Asabal pig that has a mutation in PCSK9, and it develops high cholesterol levels, high amount of plaque in the vessels, and we want to test our antibody in this and look at the effect on cholesterol uptake in the liver and smooth muscle cell differentiation in the plaque. I hope this wasn't a big shift to what we are talking about today. But please, if you have any questions, feel free to ask, and I'm happy to talk about the other project with you.
Questions for Dr. Shehadeh?
It's really nice work. So it looks like the osteopontin antibody, if I understand right, it's kind of multifactorial in how it's having an effect. So it's working in the plaque directly and is also increasing LDL uptake. Is that correct?
Right, and osteopontin has been, or let's say the knockout of osteopontin has been reported by, there's a plethora of literature showing that it reduces inflammation, reduces fibrosis. So the hope is that, or the hypothesis is that it will be multifactorial, like you said, it will help clear cholesterol from the plasma, reduce inflammation, reduce fibrosis.
Do you have any idea, like, with the effects that you're seeing, what might be most important? Like, which of those effects might be most important, or are they?
I think from conversations with the cardiologists at work, the belief is that cholesterol is it. If you reduce LDL cholesterol from the circulation, that is the most important thing you want to do.
One more, I'll have it. Have you looked at bone at all to see if it's having any effect on bone remodeling?
You mean with the osteopontin knockout?
Or the antibody? Or
with our antibody doesn't detect the mouse, so we didn't do the injection with the mice. But there is a paper on osteopontin knockout effect on bone density and hematopoietic stem cells, and I think there is an effect. It does. So total knockdown of osteopontin long term is never good. You're knocking down any gene, it's non-physiologic. So it does have an effect, a negative effect.
Other questions? So I was just curious about your that monoclonal antibody. That seems like it was. I guess, could you take us through the development of that? I mean, how were you able to target like a specific epitopes to create?
Right. So the way you develop a monoclonal antibody, you pick a region of the antibody of the protein itself. So osteopontin has RGD binding domains where it binds integrins. It has a domain that binds CD44, has a heparin binding domain, has calcium binding domain. So you get to pick your favorite area, your favorite region, and you can outsource this. And this is what we did. We send the sequence to a company. They inject that in mice, develop the antibodies, and they basically send you back the clones, and you test them, and you decide which one, based on the functional assay or the binding, which one is your favorite.
So do you have any speculation why the male and the female mice are different?
Yes, I think it has to do with beta adrenergic signaling, cyclic AMP. This is our latest data.
Yes.
So beta adrenergic signaling is these are beta receptors that are the transmembrane proteins that are famously known as targets of drugs like beta blockers for heart failure and or beta 2 agonists for asthma. These important receptors regulate the generation of a second messenger called cyclic AMP. And this second messenger then regulates a cascade of events. So we published with our collaborators at NOVA that when you knock out osteopontin, there is more cyclic AMP. There's another PNAS paper that showed the same in osteoblasts, we showed that in cardiac myoblasts. So less osteopontin, more cyclic AMP. So we went back and collected tissues between the wild type and the osteopontin knockout, males and females, and we found changes only in the female tissues. So we hope, this is going to be the mechanism explaining why there's a gender difference. Again, now, why is it regulated in the females more than the males? Many unanswered questions. But there is something definitely going on there differently in the females and the males. If the females have more cyclic AMP or more response to osteopontin knockout, and cyclic AMP is being protective, then that could give us an answer.
So how is the antibody that you generated? They're binding to osteopontin and preventing it from binding to whatever receptor?
So we thought, there's a lot of also unknowns here. It's counterintuitive. We thought when we were focusing on the kidney disease, where there is more cholesterol uptake in the kidney, and when you knock out the osteopontin gene, there is less receptor, the Ldl receptor, there is less cholesterol uptake. We said, okay, we're going to make an antibody against osteopontin. It's going to block the uptake of cholesterol in the kidney. And it turned out to do exactly the opposite. And think about it, it's totally different when you knock out the gene, you're preventing the protein from happening, versus you're adding an antibody from the exterior that's binding to the receptor and sort of stabilizing it. So bottom line, the osteopontin antibody increased the cholesterol uptake, which makes it useful for the liver and not the kidney. And so
do you know if it, because I'm thinking, oops. I was just talking to Josh here. Osteopontin is highly upregulated in metastatic melanoma.
yes.
So if you were to use this antibody, do you think you would be able to prevent metastasis?
So one of the things I learned the hard way is that do the animal work first, and before you do this, the many in vitro work following your hypothesis, because things can work differently. So the only way we would do is we tested, and with cancer, especially if it's a human cancer cell line that we can engraft in a mouse, for example, this would be an easy experiment. Inject it with the antibody and see what it does. Now, it could be bad if there is cholesterol in that mouse, and it's going to make more cholesterol go to the tumor, but it could prevent metastases, it could prevent proliferation, it could prevent fibrosis. So, and the net effect, I mean, you can't guess until you try.
You haven't.
No.
Let's try.
Yeah, okay, sure.
So the microRNA work was very fascinating, and as you said, it actually prompted you to look at some commercial prospects as well. It seems like the main barrier there is selective targeting and retention. So any thoughts on how that could be, because that seems to have an sort of an engineering angle as well.
Right.
So any thoughts on any thoughts on, or did you get some insights on that?
Right. So what got me first really interested in osteopontin is that it is for targeting purposes, and this is where we started our aptamer work. So osteopontin in a healthy organism is expressed at the minimal. If you do qPCR on a healthy heart, you barely see any transference. The only expression you would see is in the kidney and the gallbladder and the bone. However, when there's inflammation, it skyrockets. It shows like at the top of microarrays for kidney disease, for cardiovascular disease, for cancer, for neurological diseases. So the thought was, and classically people follow the receptor to deliver a drug specifically to a specific tissue. However, if you target a ligand that's going to go and bind to the inflamed organ, it's sort of, you're thinking, it's like you're ending up with the same geographical location. So osteopontin is secreted by inflamed tissue, secreted into the plasma, and then goes back and binds only on the activated receptors of the inflamed tissue. So we started working with osteopontin aptamer, which are super easy to work with. It's a sequence, you buy it, and the cancer people developed an osteopontin aptamer against breast cancer. It's a sequence, you can conjugate it with an antimer, with a mer, what have you, and it should go to where the receptor is expressed, osteopontin binds. So when we started these experiments in a TAC model of heart failure, you have to start with your controls first. So we did the TAC model, we ligated the aorta to cause heart failure, and then we injected the osteopontin aptamer alone. And then the idea was we're going to inject the osteopontin aptamer conjugated with the mer. The osteopontin aptamer alone cured the heart failure, and we published that paper in Cardiovascular Research. So we didn't even, it's much easier to cure a mouse, let's put it this way. But yeah, the osteopontin, whether an antibody or an aptamer, can itself be used for targeted delivery. Like if you want to target, you can conjugate an oligo or a small molecule to an antibody. It's been done, and then it will go specifically to where osteopontin is done, and hopefully deposit by endocytosis the cargo into the cancer cells or the sick cells. But then there could be much more, innovative ideas from the bioengineering folks, like nanoparticles, or the microRNA field is waiting for an answer. And all RNA therapeutics actually are in need for targeted delivery approaches.
I have a question about the confusion between your female and male animal.
Okay.
So, is there any difference between the basal level of osteopontin in female and male animals that when you knock out these has like kind of
Good question. I haven't looked at it. I mean, I would have to look at the ApoE mice themselves because in the healthy animal there isn't much at all. But with the ApoE, we would have to look which one is higher. I mean, classically, it's known that the females develop plaque more than the males. I don't know what that means in terms of osteopontin. But
yeah, like in the case of like let's say that's bone diseases, so like the female patients are more prone to like bone diseases, so maybe this kind of correlates with your study.
they're all linked.
Yep.
It's a mystery that we should solve. Yeah.
Other questions? If not, it's again a great pleasure to have Dr. Shehadeh give the talk, and I think it's very ironic that we had discussion on bone markers that were bad for cardiovascular disease, and in some cases, bone markers can do good things too, I guess. So, all right, well, thank you very much. And I think we're gonna segue into a panel discussion. Are we doing that right away, Josh? Or
We had it scheduled at 3, but we can take a short break.
we can take a short break, but don't go anywhere. All right, thank you.
There's coffee back here.
Coffee.
Hope I didn't take much.
Yeah,